Chapter 8, the Muscular System. Muscles make up about half of your body's mass. Muscles can transform chemical energy. We mentioned ATP before. This is energy produced from breaking down your food. So this chemical energy is changed into mechanical energy, which is the contraction of that muscle. If you've read your chapter, you saw that there are new vocabulary words, and since we're dealing with the muscular system, you were introduced to some roots of words: myo, maze, and sarco, and we'll see these roots of words when we look at terminology as we go through this chapter. There are 3 types of muscle tissues. We mentioned this before also: skeletal muscle, cardiac muscle, and smooth muscle. Now for this chapter we're going to focus mainly on skeletal muscle, but we will talk a little bit about cardiac and smooth muscles because we are talking about the muscular system. But we get into more detail when we talk about the cardiac muscle, when we talk about the heart later on, and smooth muscles when we talk about internal organs. What are the functions of the muscular system? For movement or locomotion, also to move blood through the blood vessels, food, urine, lymph, to maintain posture and body position, also to stabilize joints, to help when there's an injury to a joint, if there's like ligament injury to the joint and the doctor's concerned, they'll send the patient to physical therapy to strengthen the muscles to help stabilize the joint. The muscle helps to support and protect soft tissues. It guards entrances and exits by regulating what is allowed to move from the internal to the external. It helps to generate body heat during contraction. Contraction is doing work, and anytime you do work, heat is produced, so it helps when we shiver. It helps to maintain body temperature by producing heat. We have skeletal muscle, and before we look at the structure of skeletal muscle, we need to look at the outside, which has connective tissue. There's a layer of dense connective tissue overlying that muscle, and it is called fascia. Now fascia is unique in that it is continuous with the tendon, and we'll look at that in just a minute. And then broad sheets of fascia forms the aponeurosis. Now on this figure here, this is a broad sheet of fascia, and it forms aponeurosis. This is for muscle attachment. You have a broad sheet on the top of your head and over your abdomen, a pretty broad sheet that runs down the side of your leg for muscle attachment, and it overlies some of the muscles also and separates one muscle from another. So fascia, tendon, and aponeurosis, those dense connective tissues. Now here's bone. Now remember, when we talked about bone, we talked about a dense connective tissue on the outside of that bone called periosteum. The periosteum of that bone sort of binds with the dense connective tissue that makes up this tendon. Now, as a review, we've mentioned before that ligaments will bind bone to form a joint and hold those 2 bones together so you don't have dislocation, so bone to bone. Tendon is a connective tissue that attaches muscle to bone, and that tendon actually weaves into the periosteum of that bone. So if you had this tendon tear away from the bone, it's probably going to bring some bone with it because it's fused to the periosteum of that bone. If there's a tear, it's usually tendon from tendon and not necessarily tendon from bone, because this is a very strong union between the tendon and the periosteum of that bone. They're like continuous with one another. And then the tendon gives rise to the covering of the muscle called the fascia. And it then gives rise to several other connective tissue that covers different parts of the muscle. So connective tissue that covers each level of organization of the muscle. Now, that fascia gives rise to the other connective tissue that covers different parts that you're not going to be responsible for, but I'm going to go over this real quick so maybe you'll understand what this drawing is all about. The muscle as a whole is covered with fascia connective tissue, and then each muscle has thousands of fibers all the way down here, filaments. And as you go from the largest to the smallest, muscles give rise to fascicles, and fascicles are covered by fascia or connective tissue. And then fascicles are bundles of muscle fibers, and each muscle typically has thousands of muscle fibers, and these are made up of myofibrils, and then thick and thin filaments we'll talk about in just a minute, actin and myosin. Now, each muscle is innervated. This yellow line represents nerves, axon of a motor neuron. So there are nerves that stimulates this muscle into action, and you will have an artery for each muscle and one or more veins because it needs a constant blood supply. It will have, arteries, we'll learn this later on, but arteries give rise to arterioles, which are a little bit smaller, and arterioles give rise to capillaries that are a little bit, that are smaller and smaller until they're almost hair in thickness, very, very small. And in a muscle, these capillaries will be sort of slinky because they're wrapping around these myofibrils and deep into the filaments, because all of these structures require a blood supply. Now let's look at the structure of skeletal muscle fiber. Sarcolemma. Remember I said that there is a prefix to this word, sarcolemma. Sarco has to do with a muscle. Now sarcolemma is like the cell membrane, and here's the nucleus, and in muscle cells they are said to be multinucleated, meaning that each cell has more than, or fiber has more than one nucleus. So this membrane here is called sarcolemma, and there's that muscle fiber nucleus. Sarcoplasm is almost like cytoplasm. Remember we said cytoplasm is all the contents of the cell between the cell membrane and the nuclear envelope, but sarcoplasm is all the contents here. It's like cytoplasm right under the sarcolemma. Sarcoplasmic reticulum is this network of tubules. It is a delivery system for substances needed by these fibers so things can happen. One of the main things that it delivers is calcium. For your muscles to contract, you need calcium. That calcium and other substances are delivered by the sarcoplasmic reticulum. It's much like the endoplasmic reticulum, but it is for a muscle fiber. So this mesh of network of yellow looking tubes here represents sarcoplasmic reticulum. The transverse tubule is right here. It is in the center of these other two tubes, and these are called the cisterna of the sarcoplasmic reticulum. So it's like this. I said this was a transport system, and things dump into these cisterna, and so they can be transported. And there's the transverse tubule, the two tubes of the cisterna and the transverse tubule are sometimes referred to as the triad. Okay, it is a delivery system for things needed by this muscle fiber. Myofibril. A myofibril is a bundle of fibers, thin and thick fibers, called myosin and actin, these thick and thin fibers. So this bundle right here is called a myofibril. Okay, here's another one and another one. We're going to be looking at a model when we go over this in the lab that represents these myofibrils, and the model looks very similar to this drawing. So the myofibrils have thin and thick filaments called actin and myosin. Actin and myosin filaments play a role in muscle contraction. Okay, then a sarcomere. Now a sarcomere is a section of this fiber from this Z line to this Z line, and we'll talk about that again in just a minute. A muscle fiber will be one sarcomere attached to the next sarcomere attached to the next sarcomere. Okay, so we've gone over all the things. Let's go over all the things in this drawing again. There is the sarcolemma, that's the outside membrane around this fiber, this muscle fiber. Right under the sarcolemma will be the nucleus, and then sarcoplasm right under the sarcolemma is equivalent to cytoplasm in a cell. This network of tubes here, transport system, sarcoplasmic reticulum, and they attach to the cisterna of the sarcoplasmic reticulum. So things are dumped into the sarcoplasmic reticulum and then returned to the cisterna, and then the transverse tubule. You'll see openings to the outside, the ducts, the openings of the transverse tubule in that sarcolemma. And the three together, the cisterna of the sarcoplasmic reticulum, the two, and the transverse tubule make up the triad. A bundle of the filaments would be the myofibrils. There's a myofibril. Okay, and the myofibril have the thick and thin filaments, actin and myosin. And then a bundle of these tied together by these Z lines, or Z discs as some books call it, from this Z to this Z would be a sarcomere. Now in this drawing you see mitochondria. Now remember when we talked about cell parts and their functions, mitochondria produce ATP from the food that you eat, from mainly glucose, that provides chemical energy that these network of mechanical energy by contracting that muscle, or from the chemical energy to mechanical energy by contracting the muscle. In order to supply the ATP, you need a lot of mitochondria, because mitochondria is the place where most ATP is produced. Now, in a cell, the cytoplasm can produce a little bit of ATP, but most is produced by or produced in the mitochondria. Now, I said that this would be a very vascular tissue. It has a lot of blood vessels, a lot of capillaries. Well, that mitochondria, in the mitochondria, you need oxygen. And oxygen in the mitochondria, with the mitochondria, play a role in producing the most ATP. So the most metabolically active that muscle is, depending on where it is in the body, skeletal muscles are very active, so you're going to have a lot of mitochondria because you need a lot of ATP. Okay, sarcomere. Here's the Z line, and there's a Z line. So this bundle, this myofibril from Z line to Z line, is called a sarcomere. So I like to say the sarcomere is from Z to shining Z. Okay, Z line to Z line. It looks just like that. And as you looked at the micrograph in your lab manual, you saw a striation of muscle fibers. You can actually see the Z line in that micrograph. Okay. Attached to the Z line are going to be thin filaments actin. This purple line right here is actin. There's actin attached to the Z line. Here's more actin, 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 actin. Okay, short, thin filaments. This thick filament here is called myosin. So the thick filament is myosin. There's another thick filament of myosin. Thick filament of myosin. Okay? Actin is the thin, myosin the thick. Now, we need to recognize different areas to understand how a muscle contracts. We need to name the different areas of this sarcomere. Okay, now this is again the thick filament. What was it? Myosin. The thin filament actin. From this thick myosin, the end of this thick myosin to the end of this thick myosin, this area right here is where the Z runs through the middle, is called the I band. Okay? So the Z runs through the I band. The Z runs through the I. The Z line runs through the middle of the I band. The A band goes from one end of the myosin to the other end of the myosin. Now the A band is going to be in a relaxed muscle. Just think about this, okay? You're using that microscope, and the microscope works with light shining through the specimen. Alright, if light is shining through the specimen, if you have a layer of this thick myosin and then a layer of this thin actin, in this area right here, you're looking through a very thick layer of tissue sample. So it's going to appear dark because you're looking through both the actin and the myosin. Now in the middle here, it's just myosin that you're looking through. See the actin ends. There's the ends of the actin. So you're just looking through the myosin as that light is shining through that sample. Okay, so you're going to have medium darkness here. And then over here you're going to have another dark striation. So we say that skeletal muscle is striated, and that gives the striated appearance, is this light band. And by the way, these bands are named according to the amount of light that is allowed to pass through them in a sample on a slide. Okay, so right here where that Z line is a dark Z-shaped line. There's only actin, so a lot of light can shine through this. So you can have a real light band of light coming through here in this I band. So from the end of the myosin to the end of the myosin is going to be the A band. Again, the A band is going to have a real dark stripe and a real dark stripe with a medium dark in the middle. Dark, medium dark, and then dark again. 2 layers, actin and myosin, which is myosin in the middle. So that gives it that striated look. Okay, so the A band from the end of the myosin to the end of the myosin, the A band from here to here. Now in this relaxed muscle, from this end of the actin to the end of the actin is called the H zone, that medium dark stripe or striation. Okay, that is called the H zone. So the I band has the Z runs through the I from this end of the myosin to this end of the myosin. We just see the actin there and the Z line, so it's going to be the lightest of the striations in color. Then dark, medium dark, and then dark again. Okay, so the I band from this end of the myosin to this end of the myosin with the Z running through the middle, and just the actin is all that light is going to have to shine through. A band from this end of the myosin to this end of the myosin with this in the middle here, no actin, so it's going to be a little lighter in color. Here a little darker because the light is shining up through the actin and the myosin. So that gives that striated look when we're looking at the slide. Okay, Z line, the M line. Now the M line runs through the middle of the myosin. So you can think of M as the middle of the myosin, the M line. Now you don't have to remember this, but there is a little protein filament called titin that holds these myosin filaments in their place, and they attach at the M line. Okay, so the M line runs through the middle of the H zone. It's actually the middle of the A band too. Now we're going to talk a little bit more about these areas in just a minute. We can look at this, let's look at this picture and just review just a minute. There's the Z line. Attached to the Z line, or the Z disc as some books call it. Remember, this is a myofibril, and these are the filaments that run through that myofibril. And attached to the Z line is the actin, the thin filament, protein filament. The thick protein filament is going to be the myosin. Okay, so from this end of the myosin to this end of the myosin, it has a Z running through the middle, is going to be what? The I band, yes. From this end of the myosin to this end of the myosin, A band. Now, running through the middle of the A band is this area, and as the light shines through it, you're only going to see, it's going to be lighter in color because the myosin is the only thing that you're seeing, or the light is shining through. This area is called the H zone. And then the M line would be in the middle of the H zone, in the middle of that myosin. Okay, and what was it called from Z to shining Z, or Z, the Z line to the Z line? Sarcomere. Now, I said earlier that each muscle is innervated. It has nerves. Now, we'll learn this when we talk about the nervous system, but at the end of a nerve fiber, this is a nerve fiber, it's called an axon. At the end of the axon, there are going to be what we call synaptic knobs. Okay, in the synaptic knob, there are going to be mitochondria. Again, it's metabolically active. Energy is needed for things to happen here, so ATPs are needed here. So you're going to have anywhere ATPs are needed, anywhere that energy is needed, ATPs, you're going to have mitochondria. In that synaptic knob, you're going to have what is called synaptic vesicles. The synaptic vesicles is going to hold what is called a neurotransmitter. A neurotransmitter is a chemical, and that chemical will cause something to happen. So these are little storage containers, these synaptic vesicles, and they hold chemicals, and these chemicals make something happen here when they're released. Okay, so the neuromuscular junction, neuro having to do with the nervous system, the neuron, the fiber of a neuron, the axon comes in contact with the muscle, neuromuscular junction. Okay, a motor neuron, the axon of the motor neuron, the synaptic vesicle, synaptic knob with the synaptic vesicles. Okay, these chemicals are the way that the nervous system communicates and causes things to happen with the muscle. Now, this area is called the motor end plate. Okay, the motor end plate. You can see it here. Here's another motor end plate. It's the surface area on this sarcolemma that you have the junction of the nerve fiber with the synaptic knob with the sarcolemma, where things are going to be initiated, things are going to happen for this muscle either not to contract or to contract. Okay, so the motor end plate. Now on the motor end plate, look at this. You have the folded sarcolemma. Remember, the sarcolemma is like the membrane, okay, the cell membrane. Any time you see folded tissue or a folded membrane, why do you suppose you would have folded tissue? Pretend that you have a thread that is lining these folds. Okay, just follow along these folds. Now stretch it all out. Stretch that thread out. It's going to be really long. So look at this little bitty area, but yet when you stretch out all that folded tissue, it's going to be long. So folded tissue increases surface area where things can happen. Okay, so anytime you have folds, later on we'll look at the digestive system. In your intestines you have villi, which is a folded membrane, finger-like projections in the intestines to increase surface area. Okay, in the folded sarcolemma, you're going to have receptors, and these receptors are going to accept these chemicals, and it's going to cause things to happen. So, okay, the motor end plate has this folded sarcolemma. Okay, the motor end plate. Now, this is the synaptic knob that has the synaptic vesicles that have the mitochondria. What are mitochondria used for? energy. There's, oh, in the sarcolemma, you're going to have mitochondria too, because you need a lot of ATP, a lot of this currency for energy, the release of energy, ATP for the release of energy. Okay, so in this area here is actually sort of a cleft. A cleft is a space. Now you have the synaptic knob, you have the sarcolemma, the folded sarcolemma, and you have this space. It is going to have like a conductive gel in it, the synaptic cleft. And this conductive, these bioelectrical impulses will be transmitted in this conductive gel in this synaptic cleft. It's a space. It also allows for the movement of this chemical from when they're released from the synaptic vesicles out of the synaptic knob, the end of the axon, through the synaptic cleft, through that gel, and to the receptors found on the folded sarcolemma. And depending on what kind of chemical is released will determine what happens with this muscle, whether it's going to contract or not contract. Now the main neurotransmitter for muscle contraction is called acetylcholine. So because it's going to contract the muscle, we call it excitatory. Oh, when you get excited, things are going to happen. And for muscle contraction, excitatory. If the neurotransmitter is inhibitory, it keeps the muscle from contracting. But acetylcholine in skeletal muscle is excitatory. So whenever acetylcholine is released, then things can happen, and the potential for a muscle contraction will be greater. Now here is a animation that you can watch on Blackboard, and it gets a little bit technical. Here is the axon ending with the synaptic knob. Here are the synaptic vesicles. And in the synaptic vesicle, you're going to have acetylcholine. Now we're going to play this, and I'm just sort of going to talk you through it, though you can watch this over and over again on Blackboard. You can also watch this on the textbook website. Go to, pull down the chapter, chapter 8, and click on animations and choose function of the neuromuscular junction, and you'll see this animation. It may play a little bit better on the textbook website because you're playing from their server instead of the school server. Okay, here we go. Now what's happening is this is a action potential is a nerve impulse. That bioelectrical impulse is moving along the surface of that axon. Now what happens is as that impulse comes along, it opens this floodgate, and this floodgate allows for calcium to come in. This cannot happen. Neurotransmitters cannot be released unless there is a presence of calcium. As the neurotransmitters are released, they go into the synaptic cleft. And remember I said on the sarcolemma, the folds of the sarcolemma have receptors for acetylcholine. When acetylcholine attaches to these receptors, it allows for the influx of sodium. It causes another nerve impulse on the surface of the sarcolemma, and that impulse later on down the line allows for the release of more calcium and allows for the muscle to contract. Now what's happening here is an enzyme was released called acetylcholine esterase, and acetylcholine esterase breaks down the acetylcholine. Now if you have calcium and you have acetylcholine present, then this muscle could continue to contract. But we don't want our muscles to contract continuously. If that muscle contracts continuously, that would be like a Charlie horse. If you ever had a Charlie horse in the calf of your leg or in any other muscle, that's when that muscle contracts and it hurts because it just stays and stays and stays. It's supposed to be a voluntary muscle contraction, but it's not. It's staying contracted. That acetylcholine is not being released. You need that acetylcholine to be released so that muscle can relax. So the acetylcholine is released. It's broken down into acetic acid and choline. It is reabsorbed by the synaptic knob and recombines in the synaptic vesicle for acetylcholine again, so it can be used for the release when there's a nerve impulse and the influx of calcium, the release of the acetylcholine to attach to these floodgates to allow the sodium to come in. Everything happens all over again. So as long as there's a nerve impulse, calcium, acetylcholine, and the influx of sodium, so you have to have enough sodium here also, then you can have a muscle contraction. I'm going to end this section part 1 here with the neuromuscular junction. Next time we'll look at how the muscle contracts, and we will be looking at the sliding filament theory.